Hello again. Welcome to the FDA Grand Rounds, Pharmacovigilance in Practice from Safety Signals to Regulatory Action. My name is Ayaan Ibrahim, and I'll be introducing our speakers. We have Lieutenant Commander Anne Tobenkin, who serves as the safety evaluator on the neurology team in FDA's Division of Pharmacovigilance, DBV, within the Center of Drug Evaluation and Research. With over 20 years of specialized experience in post-market safety surveillance and evaluation, she plays a critical role in protecting public health through comprehensive drug safety monitoring. Lieutenant Commander Tobenkin earned her Pharm.D from Albany College of Pharmacy in 2003 and a Master's of Science from the University of Maryland in 2022. She began her career as a primary care pharmacist at the Veterinary Affairs Hospital in Washington, D.C., before joining the FDA in 2008. At the FDA, she initially served as a safety evaluator in the Division of Medication Error Prevention and Analysis, and then advanced to DBV, where she conducted adverse event surveillance for anticoagulants and multiple sclerosis drug products. She subsequently transitioned to the Office of Compliance, specializing in misbranding and unapproved prescription drug products. Most recently, she served as a team leader in the Office of New Drugs, where she supported the implementation and management of post-market safety initiatives, including developing and leading post-market safety report pilot program. Our second speaker is Leah Herity, who joined FDA in 2022 as a safety evaluator in the Division of Pharmacovigilance within FDA's Center of Drug Evaluation and Research, where she has contributed to multiple complex post-market safety reviews and high-impact regulatory action. She earned her Bachelor's of Science in Biological Sciences from the University of Connecticut in 2009 and a Doctor of Pharmacy and Master's of Public Health in Public Health Leadership from the University of North Carolina at Chapel Hill in 2018. After graduating, she continued her training at Virginia Commonwealth University Health System in Richmond, where she completed a PGY1 residency in pharmacy practice, a PGY2 residency in internal medicine pharmacy, and received board certification as a pharmacotherapy specialist. Prior to joining FDA, she was an assistant professor at pharmacy practice at the Albany College of Pharmacy and Health Sciences and clinical pharmacist in inpatient family medicine at St. Peter's Hospital in Albany, New York. And now I'll turn over the mic to Lieutenant Commander Anne Tobenkin. Thank you.
Well, thank you, Rakesh and Ayaan, and good afternoon to everybody, and thank you all for joining us today at FDA Grand Rounds. My name is Lieutenant Commander Anne Tobenkin, and today my colleague Leah Herity and I will be discussing pharmacovigilance in practice, from safety signals to regulatory action. Here's our disclaimer. I'll let you read that for a minute. Over the next 45 minutes or so, we will tackle the following learning objectives. To start, we'll discuss pharmacovigilance and how it's specifically conducted within our division in FDA. Then we'll go into details about 3 high-profile safety updates and share the evaluation process behind these regulatory actions. And finally, we'll share information on how to submit your own adverse event report to FDA to help with identifying future adverse events. So first, we'll start with an introduction of who we are and what we do in pharmacovigilance. Here's an abbreviated organizational chart of the FDA. We will talk about pharmacovigilance reviews conducted in the Center for Drug Evaluation and Research's Division of Pharmacovigilance, or DPV, which is circled. Notably, there are multiple offices and divisions besides ours that conduct their own surveillance and pharmacovigilance. However, this presentation will be restricted to DPV's work with marketed drugs and therapeutic biologic products. DPV is broken down into DPV1 and DPV2. These 2 divisions are further subdivided into teams that align with the Office of New Drugs therapeutic areas, such as neurology or gastroenterology. DPV teams consist of safety evaluators and medical officers. Two major concepts that we want to introduce in the beginning of this presentation include pharmacovigilance and safety signals. Pharmacovigilance is the science and activities relating to the detection, assessment, understanding, and prevention of adverse effects or any other medicine or vaccine-related problems. Pharmacovigilance centers on identifying safety signals, which is what we call those red flags that warrant further investigation. The definition of a safety signal contains a few components. First, information is derived from one or more sources. Second, there is information about a new or worse adverse event that was previously unknown. And third, there's a sufficient likelihood that the association is real to justify action to verify the issue. In 2020, CDER introduced new policies and procedures for collaborative identification, evaluation, and resolution of a newly identified safety signal, or NIS, associated with marketed drugs and therapeutic products. This map established three phases: pre-evaluation, evaluation, and action for the evaluation and management of a safety signal. The goal of this updated process was to facilitate timely evaluation and management of post-market safety signals and improve transparency with industry and the public. This figure shows our process from start to finish for signal management. I will discuss the different stages of the process, and then we will get into more details as we go through the presentation. The first phase is known as the pre-evaluation phase. This is the stage where we perform our surveillance of a variety of data sources, such as adverse event reports, literature, and applicant safety data, to identify new safety issues that may warrant further investigation. After identification of a potential signal, we perform activities to determine whether full evaluation is needed. The FDA Adverse Event Monitoring System, or AIMS, is our primary pharmacovigilance database. In DPV, we focus on post-market adverse event data from AIMS, specifically for drugs and therapeutic biologic products. Traditional sources, such as post-approval clinical trials, medical literature, specifically case reports and observational research, and epidemiologic databases, are important to obtain information on population-based adverse events. We also frequently communicate directly with foreign regulatory bodies to obtain important information concerning drug safety. Finally, we also use less traditional data sources, including social media, poison center data, emergency room cases, and toxicology assessments to get key patient-level perspectives. AIMS provides a wealth of information for post-marketing medication use within the United States. FDA receives more than 2 million adverse event reports annually. To date, we have over 20 million total reports. Every year, approximately 5% of reports are submitted directly to FDA from patients, consumers, or healthcare professionals through the voluntary reporting MedWatch process. The other approximately 95% are submitted directly from the drug manufacturer. Individuals can voluntarily submit adverse events to drug manufacturers, and the manufacturer is then required to forward these to the FDA. Manufacturers also submit solicited reports through organized data collection programs, such as patient support programs. Reports from both pathways are processed and stored in AIMS. Safety evaluators within DPV systematically review all adverse event reports submitted through both pathways, and can also perform searches on the AIMS data when specific questions arise. The AIMS database is a drug safety surveillance tool with many strengths. It includes all U.S. marketed products, and many include foreign products as well as unapproved products. AIMS includes all uses, both approved indications and off-label uses. The database includes broad patient populations. It includes elderly, children, pregnant women, and patients with comorbidities who are often excluded from clinical trials. So when is AIMS most useful? It's ideal for rare events or events with low background rates, such as acute liver failure or serious skin reactions. It's also useful for adverse events that occur shortly after exposure, such as hypersensitivity or anaphylactic reactions. There are limitations to this database, though. AIMS is susceptible to underreporting as reporting is not mandated. We're also unable to calculate incidence rates with AIMS data. It's also difficult to differentiate between worsening a pre-existing disease for which the drug is indicated, or if the event takes a long time to manifest after drug initiation. There's also frequently missing information in the reports that limits our ability to evaluate them. So now, returning to our signal triage. Once DBV has reviewed all these sources and identified a possible safety concern, how do we prioritize? Some important considerations include whether it is unexpected and serious. An unexpected event is defined as not listed in the current product labeling. Adverse events can be classified as non-serious or serious. DPV primarily focuses on serious adverse events. Serious outcomes are defined in the regulations as death, life-threatening, hospitalization, disability, congenital anomaly, or others that may require intervention to prevent another serious outcome, such as using an EpiPen after a reaction and not having to go to the emergency room. We also consider the impact on the individual, the broader health impact, and the effect on the product's risk-benefit profile. We then move to the evaluation phase. DPV reviewers use a variety of surveillance tools to identify individual case reports. We review reports and develop a case definition that meets agreed upon diagnostic features. We then assess the causality of the adverse event with the drug and then establish a case series that includes all relevant cases that describe the safety event of interest. We also use published scientific literature, preclinical, clinical, epidemiologic, and pharmacologic data to provide additional scientific and clinical information for a robust multidisciplinary analysis. After completion of DPV's analysis, multidisciplinary teams meet and agree on the next steps. Based on the findings of the evaluation, regulatory actions may be recommended based on the strength of the data, the severity of the event, and the overall public health impact. Some of the potential ways we take action and disseminate new safety information include updating the safety label. This could include adding the new safety findings to the boxed warnings, warnings and precautions, contraindications, adverse reactions, or drug interactions. Other actions include reaching out to the healthcare providers through a Dear Healthcare Provider letter, issuing a drug safety communication, or a CDER safety alert. We could also require manufacturers to complete post-marketing requirements, or PMRs, or running additional clinical or epidemiologic studies. FDA may also enforce creation or modification of REMS. FDA also issues warning letters to notify manufacturers and market sellers to prohibit the sale of an unapproved product. And finally, FDA's strongest action is market withdrawal of the product. So now that you have the basics of our work in DPV, we'd like to share with you 3 high-profile drug safety signals that relied heavily on spontaneous reports as well as other data sources, and that highlight the breadth of FDA's efforts in our mission to protect public health. The first signal that we'll discuss today involves a combination drug product, Carbidopa-levodopa, that has been marketed in various forms since 1975. We recently received a report in FDA AIMS describing a 69-year-old male with an 11-year history of Parkinson's. He was treated with oral Carbidopa-levodopa. He presented with polyneuropathy and persistent seizures despite treatment with 2 anti-seizure medications. His lab test revealed markedly low vitamin B6 levels, and per the reporter, no other cause could be identified to explain the patient's symptoms. Following oral B6 supplementation, the patient's seizures ceased and he was discharged from the hospital. Interestingly, the reporters proposed monitoring vitamin B6 for patients taking large doses of oral Carbidopa-levodopa, particularly if they exhibit symptoms indicative of decreased vitamin B6 levels, including neuropathy or epilepsy. After receiving this report, we reviewed the Carbidopa-levodopa labeling and determined there was no description of seizures or decreased vitamin B6 in the labeling. So before I go any further into my analysis, I wanted to provide some background on Parkinson's carbidopa-levodopa therapy and vitamin B6. Carbidopa-levodopa is the most effective therapy for Parkinson's disease and is available in multiple formulations, including but not limited to the oral and enteral formulation. Parkinson's disease is one of the most common neurodegenerative diseases of adulthood and a major cause of neurologic morbidity and mortality worldwide. Parkinson's symptoms arise because of progressive degeneration of dopamine-producing neurons and subsequent dopamine depletion. In the body, levodopa is converted to dopamine, thereby increasing dopamine levels. Carbidopa is co-administered with levodopa because it inhibits peripheral levodopa conversion, allowing for greater levodopa availability, and it also helps decrease gastrointestinal adverse reactions. Levodopa is converted to dopamine by the active form of vitamin B6, which acts as a coenzyme. Carbidopa also irreversibly binds to the active form of vitamin B6, further depleting its availability. Vitamin B6 deficiency can result in a spectrum of clinical manifestations, depending on the severity. Stomatitis, neuropathy, and depression in less severe scenarios, and microcytic anemia and seizures in the more severe settings. Seizures resulting from decreased vitamin B6 is not a new concept or finding. Other drugs, such as isoniazid, and disease states such as alcoholism, are known to decrease vitamin B6 and also result in seizures. So after identifying this report in Ames, DPV conducted a literature search for vitamin B6 deficiency-associated seizures in carbidopa-levodopa products and identified similar case reports describing refractory seizures in patients with low vitamin B6 with carbidopa-levodopa therapy. After collecting potential reports of seizures and decreased vitamin B6 from different sources, we then assessed for causality. In other words, the likelihood that the carbidopa-levodopa is responsible for the decreased vitamin B6 and seizures we're evaluating. While there are many scales and algorithms that can help us assess causality, all of these tools share some common concepts. 6 of these are listed here, starting in the first row. Temporality. This means that the exposure to the drug came before the event. Dechallenge refers to whether the event goes away when the drug was stopped. Rechallenge refers to whether the event started when the drug was restarted. We also consider the dose-severity relationship. Does a larger dose of the drug cause a more severe effect? Biological plausibility is, in essence, does this adverse event make sense in terms of the drug's pharmacokinetic and pharmacodynamic profile? Evaluating biologic plausibility requires knowledge about human biology and disease pathology. And finally, we have the idea of confounding, which is one of the most important concepts in determining a causal association. Essentially, we want to rule out anything else that could be influencing the drug-event association we're evaluating. After identifying Ames and literature case reports and assessing for causality, we identified 14 cases of vitamin B6 deficiency-associated seizures with carbidopa-levodopa therapy. Although we searched all data sources from the time of approval, we only identified cases from 2014 up to a recent publication in September 2025. We found an equal distribution of cases with oral versus enteral suspension formulation. We only identified cases that described doses of greater than 1,000 milligrams of levodopa per day for both oral and enteral suspension. We also identified 2 cases describing vitamin B6 deficiency-associated seizures after a dose increase, possibly demonstrating a dose-severity relationship. Almost all cases reported cessation of seizures after administration of vitamin B6. Concerningly, 7 cases described administering multiple anti-seizure medications, in some cases up to 4, to treat seizures prior to identifying the vitamin B6 deficiency. 2 cases had fatal outcomes with seizures, possibly contributing to the fatal outcome. However, autopsies were not completed. We also noted the biological plausibility discussed in the previous slides, showing the interplay between vitamin B6 and carbidopa-levodopa and seizures. Based upon our review of the cases and medical literature, The multidisciplinary team concluded that there is reasonable evidence of a causal association between carbidopa-levodopa products and vitamin B6 deficiency leading to seizures. Further, this safety concern was not adequately characterized in the labeling. Subsequently, the labeling for all carbidopa-levodopa products were updated to warn about the risk of vitamin B6 deficiency in seizures. The labeling describes the possible dose-severity relationship, how seizures related to vitamin B6 deficiency may not respond to traditional anti-seizure medications, and that other symptoms may also be present that are indicative of the vitamin B6 deficiency. The labeling also included a recommendation to monitor vitamin B6 levels prior to the initiation of therapy and periodically while on treatment. A drug safety communication was also issued to more broadly communicate our findings along with the labeling update. So now this concludes my part of the presentation. I'm going to pass the microphone to my colleague Leah Herity to continue with the safety signals.
All right, thank you so much, Anne, for taking us through an introduction to pharmacovigilance and safety signal management here at FDA. I'll continue our discussion with recent safety signals that have been identified and evaluated at FDA. The second signal we're going to discuss today is hyperthermia and scopolamine transdermal system. And this signal was originally identified when we became aware of a drug safety update that was posted online by a foreign regulator that was describing the risk of anticholinergic side effects, including hyperthermia, with scopolamine transdermal system, or what in some countries is called hyoscine hydrobromide patch. It's the same medication, just two different names for it. And when this drug safety update was published, in the United States, scopolamine transdermal system is already labeled for a variety of anticholinergic side effects, such as neuropsychiatric events, decreased gastrointestinal motility, urinary retention, but it wasn't specifically labeled for hyperthermia. And we noted a few other factors about this drug safety update that caught our attention. The next was that the foreign regulator had identified reports of serious and life-threatening anticholinergic side effects. And if you recall from earlier in the presentation, the seriousness of the outcome of an adverse event is something we definitely want to consider when we are triaging and prioritizing safety signals. And in fact, we saw that they had identified death in a child that was linked to hyperthermia, specifically from scopolamine transdermal system. We also noted that they had identified several cases that had occurred when the product was used outside of the license. And in the United States, we often call this off-label use. And the examples they had seen of this outside of the license use was long-term use of the product. So not just using this to prevent motion sickness, let's say for a short period of time, like on a cruise or on an airplane ride, but using it for several months on end. They also found that there were cases where patients were using it for indications other than motion sickness and where it was being used in pediatric patients. So all of these factors, in addition to others, really made us want to take a closer look at this potential safety signal. And scopolamine transdermal system has been marketed in the United States for decades. It contains scopolamine, which is a belladonna alkaloid and is an anticholinergic agent. The scopolamine is formulated into a transdermal system that is designed for continuous release following application of the patch to the skin. In the United States, scopolamine transdermal system is indicated only for use in adults, specifically for the prevention of nausea and vomiting associated with motion sickness and prevention of postoperative nausea and vomiting. It is not approved for any indication in patients less than 18 years of age, so it's not approved in pediatric patients. And all anticholinergic agents, not just scopolamine, but other anticholinergic agents as well, have the potential to cause anticholinergic side effects, or what we might call an anticholinergic syndrome or toxidrome, that is described here on the right side of the slide. And you'll see that one of the known manifestations of anticholinergic syndrome is hyperthermia. And if you recall, when we were discussing patient-level causality assessment earlier in the presentation, one of the factors that we look at when we're assessing the causal relationship between a drug and an adverse event is biologic plausibility. And so we felt that there was strong biologic plausibility for scopolamine, an anticholinergic agent, to be causing an anticholinergic syndrome effect like hyperthermia. And you can see that there are other manifestations here that are also potentially reflective of temperature dysregulation, like skin flushing or dry skin, which can occur if patients are not sweating anymore. That's called anhydrosis, and that can happen with an anticholinergic syndrome. For this signal evaluation, we used a variety of different data sources. We did use the traditional data source of Ames reports. In particular, there were a handful of cases in this signal evaluation that came directly from consumers or healthcare professionals. And if you recall earlier, we talked about the fact that reports coming from patients, consumers, or healthcare professionals directly to us at FDA only represents a fairly small percentage of the reports that we get to the Ames database for drugs and therapeutic biologics. It's only about 5%. But these direct reports are a really valuable source of information for us. It helps us see these adverse events from the perspective of the prescriber or the patient. And in this particular signal evaluation, it was really helpful for us to have these reports. We also know that scopolamine's been marketed for many years, not just in the US, but also in foreign countries. And we considered the fact that it was possible there were foreign reports of hyperthermia that had occurred in other countries that that's not necessarily required to be submitted to FDA, but we wanted to have the opportunity to review these reports. So we reached out to foreign regulator colleagues and the applicant to have them provide any potential foreign reports of hyperthermia with scopolamine transdermal system to allow us to review those. We also conducted a medical literature search to look for published case reports, and we used drug utilization data. And drug utilization data essentially gives us a context in which we can interpret the reports that we're seeing. The drug safety update from the foreign regulator had identified cases in pediatric patients, and we were seeing these reports too. So for drug utilization data, we looked particularly at off-label use in the pediatric patient population. We wanted to see how frequently is this being prescribed to pediatric patients. When it is prescribed, is this a short course or is this a 90-day supply? And also, who is doing most of the prescribing? Is it neurologists, primary care physicians, pediatricians? And so drug utilization data gives us all this information. It's essentially a foundation or a background in which we can interpret the reports that we're reviewing. In looking at all of these data sources, the team that reviewed this safety signal identified 13 cases of hyperthermia with scopolamine transdermal system. And you can see that we did have some cases in the United States, but we also had several cases that were occurring in foreign countries. We had some cases that we had received in the earlier years after the product was approved, but it was not until more recent years that we received additional cases, and in particular cases that had serious outcomes that were concerning. And although there were some cases that did appear to occur when this product was being used in a way that was consistent with US prescribing information, the majority of cases actually looked like they were occurring with some sort of off-label use of this product. For example, you can see that the majority of the cases of hyperthermia with scopolamine transdermal system happened in pediatric patients. And if you recall, this drug is not approved for use in pediatric patients in the US. We also noted that we had cases where that occurred when the product was being used for a reason other than the approved reasons of motion sickness or postoperative nausea and vomiting. The most common was secretions. An example of this would be using it to manage drooling in a patient with cerebral palsy or another neurological condition. And then we had a small number of cases that involved administration techniques that were not consistent with US prescribing information. For example, cutting the patch and applying a small piece of the patch, or applying multiple patches at once when the product should only be one patch at a time. And the events in this case series were resulting in serious outcomes. We had 4 cases that resulted in hospitalization and 2 cases of hyperthermia that resulted in patient death. And the multidisciplinary team that worked on this review determined that regulatory action was necessary to warn the public of this risk. We made safety labeling changes, and this included a new warning for hyperthermia, as well as corresponding changes to other parts of labeling like use in specific populations. This is where we can describe that this was happening in pediatric patients, and patient counseling information. We also released a drug safety communication. This provides a little bit more detailed summary of the cases where you can see the types of patient ages where this was occurring, any potential risk factors we identified, body temperature of the patients when they had hyperthermia, when that was reported. And it also discusses potential mitigation techniques. And this communication is really targeted both to prescribers who are prescribing this information or this product for patients and to patients and consumers themselves. Okay, the final safety signal that we're going to discuss is a little bit different than the prior two. This is with the prior two, of course, were with FDA approved medications. This is with an unapproved non-prescription chemical peel product that was associated with serious skin injury. So chemical peel products for this signal evaluation, these products can contain a variety of different acids. And some of the examples are shown here on the slide, like trichloroacetic acid, glycolic acid, salicylic acid, lactic acid. There are others as well, and some of the products actually contain a combination of these acids. For this signal, it pertains specifically to consumer self-application of unapproved non-prescription chemical peel products that contained one or more of these acids that would typically be used by a healthcare professional, but in this setting, were being purchased online from websites like Amazon.com and applied without that healthcare professional supervision. And these products are not FDA approved, so they have no FDA approved indications. But nonetheless, they are marketed to improve acne, improve skin discoloration, get rid of wrinkles, promote collagen production. And this signal was initially identified when safety evaluators in the Division of Pharmacovigilance noted Ames reports of serious skin injury from self-application of a highly concentrated trichloroacetic acid product that had been purchased online. But we didn't limit our evaluation just to these highly concentrated products. We also looked at chemical peel products that contain more typical concentrations of acids just to see if those products were also potentially causing skin injury. And for this signal evaluation, we did, of course, use the traditional data sources of Ames reports and medical literature. And in fact, the team that worked on this evaluation found several reports from these traditional data sources. But to supplement that information, the team also used a bit of a non-traditional source to find cases, and this is consumer reviews on Amazon.com. And they identified 3 products that had multiple consumer reviews describing skin injury after self-application. And these consumer reviews, of course, included pictures that a consumer would take after they experience the skin injury and post that picture in their review. And I think this really represents the evolution of looking for new data sources for pharmacovigilance and trying to look at adverse events from different viewpoints whenever possible. After these cases were put together, they were provided to the Division of Pharmacovigilance's dermatologist, who then assessed each case for the extent of skin injury. On the next slide, I am going to show an example picture of skin injury that happened with these products. This is a case that we identified in the medical literature. It was published by Liu and colleagues. And in this case, we had a 38-year-old male who had purchased a 50% trichloroacetic acid chemical peel product from an unspecified source. He was applying it to self-treat acne. Pretty quickly after he applied this, he noticed stinging on his skin. He tried to wash the product off, but over the next few days, he noticed worsening skin discoloration. He went to a dermatologist. The dermatologist, as you can see here, saw that the patient had erythema and well-demarcated hyperpigmentation on the face and temple that you can see where this product was applied. They did a skin biopsy and diagnosed this patient with a chemical burn from this chemical peel product. And this is just one example. Many of the cases in this case series had pictures that were provided in the reviews or in the case that showed really a wide variety of these types of skin injuries. And the multidisciplinary team that worked on this review determined that regulatory action and communication with the public was necessary. This came in the form of a CDER safety alert. This was warning the public against purchasing and using these chemical peel products without professional supervision. We also issued warning letters to 6 companies that were selling non-prescription chemical peel products that were associated with these severe skin injuries. Additionally, another method that we sometimes use to communicate with the public is through publications in peer-reviewed literature. And that is what this group did as well. They published essentially a summary of the case series for both professionals and consumers to be able to access this in the medical literature. And this can be a good way for us to reach a broader audience in our communications. Hopefully, as we've discussed the 3 recent safety signals, you've started to notice that adverse event reports, especially those received directly to us from consumers and healthcare professionals, are really important for our ability to identify and evaluate safety signals at FDA. So we wanted to spend just a couple of minutes talking about how to submit an adverse event report to FDA. Our online MedWatch adverse event submission program is a direct and reliable way to share adverse event information with us. We have different forms for a healthcare professional and a consumer. And I think the most efficient way is generally to submit this online, but we also have the option to print the form. You can fill it out and fax it to us, mail it to us, or we have a dedicated 1-800 number if you prefer to communicate the adverse event with us via telephone. And if this adverse event pertains to a drug or a therapeutic biologic, then it will come to us in the Division of Pharmacovigilance, and it will go to the safety evaluator or sometimes multiple safety evaluators that are assigned to that medical product. And to actually transmit a report to FDA, there's actually only 4 required pieces of information. These are the patient that experienced the event, the product, the adverse event itself, and the reporter. And although these are the only 4 required pieces of information, we as safety evaluators really need a lot more detail to be able to fully assess an adverse event report. And some of those details are shown here on the slide. You may remember them again from when we talked about causality assessment, information like D-challenge and re-challenge, patient characteristics like baseline medical conditions, temporality, as we discussed. So this would be information like how long had the patient been on the medication before the adverse event occurred? Were there other medications that they were receiving? And might those be alternative etiologies for the adverse event? So what we do with all of this information is we put it all together, and it helps us determine if there is a causal relationship between the product and the adverse event. And if there is that relationship, how strong is the relationship? Before we close, we just wanted to leave you with 4 key takeaways from our presentation. The first being that pharmacovigilance within the Center for Drug Evaluation and Research identifies adverse events and adverse events associated with product quality issues, definitely for FDA-approved drugs, but also for unapproved products. An example of that was the chemical peel products, as well as counterfeit products. Safety signals can be identified from a variety of sources. We have our traditional sources like adverse event reports in the AIMS database and published medical literature, but we also try to use non-traditional sources when possible as well, such as Amazon.com consumer reviews. We also have other sources that we maybe use a little bit less frequently, like interagency collaborations or communications with foreign regulators. And when we perform a signal evaluation to determine that regulatory action is necessary, there's a lot of ways that we can do this. We can make safety labeling changes, release drug safety communications, have CDER alerts, send warning letters, and then we also try to communicate with the public in other ways, such as through publishing in peer-reviewed medical literature. And finally, we are always grateful to consumers, patients, healthcare professionals who take the time to submit adverse event reports to us, in particular when the adverse event is both serious and unexpected. And again, unexpected, when we say that, we mean it's not well described in labeling for that product. If you do in the future encounter a safety issue or an adverse event with a drug or therapeutic biologic, we hope that you will consider sharing that information with us here at FDA. As safety evaluators in the Division of Pharmacovigilance, it is very valuable and important information for us to have. Thanks so much. I think that we have time for questions, and we really appreciate everyone joining in and for your interest in the topic of pharmacovigilance and post-market safety.